Good day, grade 12 learners. Welcome to today's Life Sciences lesson. My name is Samantha Ragunanan. This lesson is brought to you by the Gauteng Department of Education in collaboration with Saibono Discovery Center. You may contact us on the following number: 063 508 1301. I'll repeat the number: 063 508 1301. You may send messages but not phone calls or videos. Let us get started. Today the topic that we're going to cover is genetics and inheritance. We're going to cover the concepts of inheritance, monohybrid crosses, and sex determination. I will do the first part of the lesson and my colleague Agnes will join you a little later in the session. So let us start. So genetics and inheritance. What exactly is genetic inheritance? Basically, genetics is the study of heredity and variation. So what this actually means is that, when you hear people saying, Nisia, you look just like your mum, or Nahum, you got your dad's nose, hey? Or you look like your grandpa. So you've heard things like that, where people will tell you that you look like someone in your family, or maybe you've got features that maybe were undesirable or maybe desirable from your parents. So that just proves that life science is such a current subject. It's something that involves us every single day. So genetics and inheritance is basically studying the characteristic and the traits that we inherit from our parents, who inherited it from their parents, grandparents, and so on. So that is what's so amazing about it, is the fact that where you are sitting or where you are unique. When I say you are unique, you have your DNA is unlike anyone's in the world. So there's no one on this earth that's got DNA like you do. So let that just resonate with you. But let's get back to the syllabus and see what this involves and how it is included in the curriculum. So what you need to know is that genetics and inheritance forms 48 marks of the exam, and it's found in paper 2. So this is a big chunk of marks, grade 12s. So you have to know how to do it. You have to follow the exam guideline, the topics in the exam guideline, and know according to these points in the exam guideline what you need to know. So I'm going to touch on the first 3, and in the following weeks we're going to do the rest. Okay? So for today, what we're going to do, we're going to study is concepts of inheritance. So we're going to study the different concepts or terminology with regard to genotypes, phenotypes, homozygous, and heterozygous. I know it sounds so overwhelming, all these big words, but it's so easy when you break the words up into simpler terms, and then you will see that you actually find the answer in the question. I'm going to show you how to do this. And then you have monohybrid cross. The monohybrid cross involves a diagram and involves problem solving. But you must remember that when you are going to do the monohybrid cross, you need to know the terminology. So when I'm going through this first part of concepts of inheritance, you need to pay attention, because you're going to take that information and you're going to use it when you are doing the monohybrid cross. And then we're going to touch on sex-linked inheritance. So this is doing genetic problems involving 2 sex-linked disorders, example, hemophilia and colorblindness. Okay, so if we move on to the next slide, there is a picture of a gentleman. His name was Gregor Mendel. He was known as the father of genetics. So this guy is one of those people that I know really liked peas. No, I'm not talking about eating peas. I'm talking about the fact that Gregor Mendel was the first biogeneticist. What he did was, he was someone who lived in a monastery in an isolated area, and he studied the garden pea plant. And as he walked past the garden pea plant, he kept walking back and forth, and he realized, hey, there's something that I can observe from this. If you had to look at the seeds, what he observed is that the seeds were either round or wrinkled. The flowers were either white or violet. He didn't see any red flowers, any variations. They were either or. So if you look at the pods, now the pods are basically those long structures where inside you find the peas, okay? So when he looked at the pods, he looked at them. They were either yellow or green. So he realized there was no in between. Basically, the pods it was either a certain color or a certain texture for the seeds, or a certain height. The stems were either short or tall. So Gregor Mendel based his studies on this. His research is what we base today's genetics on. And the research has gotten extensive, but he was basically the founder of the entire program. Okay, so whatever information that he discovered, we have built on it today on an extensive level. So if you're going to pursue a career in genetics and inheritance, this is something that would really fascinate you if you're looking at doing something in bioengineering. Let us look at the concepts that we are going to cover. So now, when we're looking at these concepts, you can see here, we've got a little bubble that says here, concepts commonly confused and used interchangeably. So we didn't just find these concepts anyway. We've looked at past exam papers. We've looked at learners answering question papers and all of that. And we've realized that learners are constantly making a mistake between terms that sound similar but are not the same. So the concepts we're going to cover are chromatin versus chromosomes, genes versus alleles. You must know the difference between a dominant allele and a recessive allele, a phenotype and a genotype, and homozygous and heterozygous. And I know what you're thinking, all of these terms, how am I going to remember it? I'm going to teach you how to remember it. I think that genetics is a subject that many learners love to hate, and there's no reason to, as long as you understand the basic concepts, which we're going to cover today. So please pay attention, make notes as we're going along, so that you can remember every single thing and every single way to remember your notes. So let's go on to the first term, okay? So we're talking about chromatin versus chromosome. So there's the chromatin. Some people like to say chromatin, some people like to say chromatin. So basically this is a mass of DNA strands that is formed during meiosis, during interphase of meiosis, and we know that this takes place in the nucleus. And we can also see that the strands of the DNA, they cover histones. Can you see the histones? Now histones are proteins. And then later on during meiosis, the chromosome is formed. So you can see, first the chromatin is formed and then the chromosome. So you cannot interchange those two terms because they're two separate things. I really like to think the chromatin looks like a bowl of noodles. So when you're getting confused in the exam, just picture the diagram in your mind and know that one is an entangled mass that's surrounding proteins called histones, and then you have the chromosome, which has a very distinctive shape, almost like a butterfly, but not a butterfly. Okay? So when you're given diagrams, you'll be able to distinguish that. So let's move on to the next term. Genes versus alleles. Now I need you to really pay attention here, because this is one of the most common mistakes that grade 12s make. You know, even with some of our grade 12 life science teachers, it is a challenge. So if you're struggling with this, don't feel bad. But pay attention now and draw diagrams as I'm doing this so that you are able to navigate this term. Now, when we talk about a gene, so we need to look at the definition so that we understand what we are talking about. So when we're talking about gene, it is a segment. Segment is just another word for section or part, okay? So it is a part. It is a part of the DNA. It is a segment of DNA, a chromosome that codes for a particular characteristic. So basically a gene is a section which you find in the chromosome. So that would be the area that you would find the gene in, okay? An example of a gene would be hair color or eye color or hair texture, skin color. So you can see it's a very general term. Sometimes when you get confused between a gene and an allele, you must use examples. So now I said eye color and hair color. So you already know that it's a broader description. A gene is a broader description and it is a segment found on the chromosome. So now we're going to build up on that point and we're going to discuss the allele. Now the alleles are alternate forms of a gene. What does that mean? It means it's a variety of the gene, isn't it? It is a variety of a gene, or it's a different type of the gene. So for example, if the gene was, for example, here we said hair color. Okay? The allele is the alternate form of this gene. So the allele would be found here where you see the little pink spots there. That's where the allele will be found. It's localized on the same locus in homologous chromosomes. Now what you need to understand, this fancy word locus, it just means location. It just means where can you find it. Okay, so sometimes we look, a lot of us, English is not our first language, and then we look at words and we become overwhelmed. Sometimes they're just fancy words for simpler words. So when we're studying, if you don't understand the word, we go to the dictionary or we go to Google. You guys like Google everything. You go there and you just Google the word and then you'll understand, because you can't just learn this terminology off by heart. You have to know what it means. So like, for example, we have alternate. Alternate is a variety, that means different forms of the gene, localized on the same locus. Do you see The alleles here are found directly opposite each other. They are in line with each other. So that's what they mean by it's localized on the same locus. So if the gene is the hair color, then what would the allele be? For example, the allele would be maybe black hair or brown hair. Do you see how it works? So sometimes you won't remember the terms, but you know, hey, gene is the broader description and allele leads to the specifics of each one. We know from our definition, a gene is a segment of a DNA or a chromosome that codes for a particular characteristic. Codes for a particular characteristic means we're talking about the characteristics that remember we spoke earlier on about you being unique. There's no one in this world that looks like you because of your genetic makeup. So that's what we're talking about, your genes. Not your denim jeans, the ones that you're wearing, the genes within your body, the stuff that makes you so the amazing person you are. What we see on the outside and what is built on the inside. So we know that the gene, for example, would be hair color, and the example of the gene would be either black hair or brown hair. And then you have different types of genes. Like I said, skin color, all of those things. Okay? But let's look at the diagram that we have here. We've got the allele for purple flowers and we've got the allele for white flowers. Do you see the section where I encircled? That is where the gene is. Inside there, in line with each other, it's the allele. And then we have alternate forms of the allele. Here you have the allele for purple, and here you have the allele for white flowers. So we can't find the allele for the flower color here. It is directly opposite each other. It's in line with each other. That is something you need to remember for a little later on. So now I don't think we're going to make a mistake between a gene and allele, okay? Because we know the difference now. I hope you are making notes. I hope you're copying my diagrams at home and you are on board with this. Let's move on to the next term. Phenotype versus genotype. Now, this is, a lot of learners become confused with these terms, but it is very, very simple. Let's look at phenotype. So when we talk about phenotype, I always like to look at the word in the question, and then I like to make a story out of it. And then when we're writing the exam, then we'll remember, hey, you know, this word has these letters in it, and it triggers off a memory in another part of my brain. Okay, so let's look at the definition of the phenotype. The phenotype, this is the external physical appearance of an organism. The external meaning outside. Remember I said we need to know what we're talking about. We must look up meanings. External means the outside, the physical appearance of an organism. The phenotype is determined by the genotype. And you're wondering what's genotype? We're coming to genotype now, okay? But let's focus on phenotype. The phenotype, when both recessive and gene alleles are present. So let's talk about the word phenotype. So phenotype is the physical features. But I like to say, take the PH and make the word photo. Photo, like a photograph. Like the selfies that you like to take all the time in the class, okay? So it's the physical appearance. So when we look at the photo, we can see, or the phenotype, we can see brown eyes, brown hair, straight hairline, skin color. All of those things is a description. You can describe it in words. And the same thing with the phenotype. The phenotype is usually described in words, okay? That's what makes it different from the genotype. So we're going to talk about the genotype now. So like we say, PH for photo on the outside of physical features. Genotype is the genetic makeup. Let's see there. Genotypes is the genetic makeup. So that means what is inside, the makeup on the inside, the genetic makeup. So let's read the definition. This is the total genetic composition of an organism. It is the information present in the gene alleles. Remember, we spoke about alleles. For example, two capital B's, capital B, small b, or BB, lower caps. So you can already see the difference between genotype and phenotype. The phenotype, you express most of the time in words, and it's a description, it's an outward appearance. The genotype is what is on the inside, the genetic makeup. And how do we know if we see words and we see letters, how do you know which one is the genotype or the phenotype? The genotype is expressed in letters, and the phenotype is expressed in words, okay? But you need to know the genotype in order to establish the phenotype. The genotype is what makes the phenotype. So for example, think of the genotype as like a sort of recipe. So in the genotype is all these ingredients, okay? So you come to my house, I bake a cake. So the cake would be the phenotype. You see what is on the outside. Do you know the ingredients? No, maybe you can guess, but you don't know the ingredients. The genotype is the list of ingredients, the genetic makeup that is on the inside. So do you see the difference between genotype and phenotype? And I'm sure you've been paying attention to the picture of Brad Pitt on this diagram. There's Brad Pitt. And let's see if you know now. So this one, the picture of Brad Pitt, those of you who don't know, Brad Pitt is an actor. And if you look at the picture, you can see that he's got brown hair. He's got, I think it looks like green eyes, and he's fair, his complexion is fair, and all of those things, nose shape, all of that we can see. But this would be obviously, remember, photo, picture, outward appearance, what would this be? Genotype or phenotype? It would be the phenotype. Yes, very good. Well done to those who got it right. And then the genotype, you can see, is this is what Brad Pitt looks like on the inside. Not very pretty, hey? But in any case, that's what his genetic, that's what his DNA in his body looks like. You've come across this diagram. You know this diagram because you did DNA in term 1. And remember, DNA is very closely related to this topic. Okay? But we're going to unpack that a little later on. I don't want to bombard you with too much of facts. Let's just learn the definitions and know what each definition is so that we don't interchange or make a mistake with the definitions, okay? Homozygous versus heterozygous. Okay, so let's see. Before we even look at the content, let's look at the word. Let's break the word up so that we know what it means. Homo. What does homo mean? Homo means the same. Zygous, come on, you just did human reproduction. Zygote. So obviously we know that same, and we know what are zygotes. Are they diploid or haploid? Yes, they are diploid. So I can already see something is going to be the same, and it's got to do with something that is diploid. And then we talk about heterozygous. Hetero, obviously, if this is the same, then hetero means different, okay? And zygous coming from the previous definition. So now that we got that at the back of our mind and we know the words a little bit, we've orientated ourselves with the words, let us go through the diagram. So there we have diagrams of diploid cells. Now, you know, I keep saying diploid, 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 but that's important because we need to know that they've got the full set of chromosomes from their parents. That means they got one from mommy, one set of chromosomes from mommy, and another set of chromosomes from daddy. So that we don't look like clones of our parents. Some of us want to, some of us don't want to, okay? So that's what makes you look similar but not different. Different but a little similar to your parents, okay? So let's look at the diagram. So here we have two alleles coding for the same gene. So let's take, for example, capital B, maybe it's black, okay? So there's the gene and there's the alleles on them. So we can see already this is homozygous. Why? Because the letters are both capital letters. Now here the secret is look for which are both capital letters or which are capital letters and small letters, because the minute you see, it's common sense. If you see a capital letter and a small letter, they're different. So it's obviously going to be heterozygous something. If you see the two same capital letters, it's going to be homo. So there's a trick to it, hey? So let's see here. So there you have the gene and you have capital B, and that would be homozygous. And then in the next diploid structure, we have got capital B and small b. Both of them, even though you have different letters, they are coding for the same gene. And you got capital B and small b. Are they same or different? They are different. So we make them heterozygous. See how easy it is? And the next one, we've got homozygous. Even though we've got two recessive genes, two recessive alleles, they are homozygous. So that is the difference between homozygous and heterozygous. It's basically looking for the letters to see if they are same or different, and also if they are lower caps or capital letters. So this all you should be taking notes of and make. These are all study tips. You don't just get them in the textbook. These are tricks that you need to learn so that when you study the notes, it's not so difficult. We're going to move on to the next term, which is dominant and recessive alleles. So now let's talk about dominant and recessive alleles. So the word dominant already, I always like this. Dominant is when something overpowers. It's like it just takes over. Like if you're standing with your friends and you're talking in this little quiet voice, I think we should go play tennis today, and your friend comes in a loud voice, no, we need to go play soccer today. So that person is dominating, they're taking over the conversation, and what happens? You eventually go to play soccer because they're so loud, they're dominating, they're taking over everything. So the other one is masked, isn't it? But let's talk about dominance. So a dominant allele is an allele that masks. What does it mean when it masks? This actually explains what a dominant allele is. When you wear a mask, it covers what is in the back, isn't it? You can't see the face behind the mask. So the same way, an allele masks or suppresses the expression of the allele partner in the chromosome pair and in the dominant characteristic. And the dominant characteristic is seen in the homozygous and heterozygous state. So you can already see there is the genotype capital T, and capital T, small t. So what they're saying is that that capital T is going to dominate. That means that tall allele is going to overpower the short allele, and the offspring is mostly going to be tall. So let us see how this shows in the diagram. So we always start with the genotype because the genotype is what actually informs. The genotype comes first and then you see what the outside is. So the genotype is wrinkled recessive. The recessive is that person with a tiny voice, the one that's not going to be heard. And then you get that loud, dominant voice, and that is round. So when we are writing dominant, because we're using the example of a loud person, the volume is high, so it's going to be a capital letter. These are just ways you can remember it. You can use your own strategies. Okay, so that is the dominant. The round is the dominant, and we express it with a capital letter. The recessive is the quiet voice. It's always going to be masked. So that would be the small letter. Okay. So as we can see, we got the dominant allele and we got the recessive allele. And what does the dominant allele does? It overpowers the recessive allele. And what is the phenotype? It's going to be round. There you can see the diagram. It's round. Okay, so that's how it works. The same principle is applied to the recessive allele. So let's see. We've already discussed the recessive allele, but we're going to go through the definition so you can become familiar. Because remember, you can be asked this in biological terminology, even in multiple choice questions. Okay, so we need to know the definitions. When you study the definitions, understand what it means, but you need to know them as it appears in your notes. Okay, because sometimes when we write it just the way we understand it, we can leave out important information, and then we can miss out on the second part of the mark. So an allele is suppressed. That means it's sort of masked when the allele partner is dominant. The recessive trait will only be expressed or seen in the phenotype if both alleles for the trait are homozygous recessive. That means when there's no loud dominant allele there to take over, when there's just the two recessive alleles, then they are expressed in the offspring. Then you can see, like look at this example. There we got the two wrinkled recessive, the two small r's, and you see that P? It is wrinkled. Okay, so that is when it's expressed. So don't always think that the dominant allele is always gonna be in the offspring. If you've got two recessive alleles, homozygous, it can be expressed in the offspring. Okay, let's move on. Now, Mendel's law of inheritance. Now, just like many theories, whether you are doing maths or geometry or any other theory, you need to understand. You can't just learn the theory off by heart. Like Mendel's laws of inheritance, you can't just learn it off by heart. You need to understand what it means. So I'm going to do a little example, and then we're going to read the law together, and then it will make more sense to you. It says if an organism has two different alleles for a trait, okay, only one is shown. So the dominant allele is fully shown, the recessive allele is masked by the dominant allele. So there you have the dominant allele Y, and you have the recessive allele. So out you got the dominant allele and the recessive allele. Only one is going to show in the offspring. Let us see what Mendel's law of dominance says about it. So Mendel, if you remember correctly, he was the guy who was studying peas. He was observing the pea plants, and he based his studies on this. So he introduced these laws. Quite amazingly enough, this was in the 1860s, and these laws are still used today. So he was quite an intelligent guy. The law states, when two homozygous organisms with contrasting characteristics are crossed, all the individuals of the F1 generation will display the dominant trait. So that's like a whole lot of information, but let's break it down. Two homozygous organisms with contrasting characteristics. So if it's homozygous, it's going to basically be a cross with a dominant characteristic and a recessive, because we speak about contrasting characteristics. So if we speak about, let's just say B is homozygous times BB is recessive. So that would basically maybe be a black rabbit crossed with a brown rabbit. If they are crossed, then all the individuals in the F1 is not racing. We're not talking about racing now. We know you like Grand Prix, but we're not talking about racing. F1 is basically the offspring. But if you can't remember that in the exam, remember P represents the parents and F is the family. So the family, you have the mother and the father, they form parents, isn't it? And later on when they have offspring, they form a family, F1. That's one way to remember it. So an individual that is heterozygous for a particular characteristic will have the dominant trait as the phenotype. So what they're actually saying here is that this is going to be the phenotype. So we have contrasting two different characteristics, they're crossed with each other. But the individual that is heterozygous for a particular characteristic will have the dominant trait as the phenotype. It's what we spoke about earlier on, how the dominant trait masks the recessive trait, okay? Let's move on. So this is another law of Mendel. It's Mendel's law of segregation, okay? Now, when you do Mendel's law of segregation, you need to know what is segregate. What does it mean? It means to separate, segregate, separate, segregate, separate. You must remember that. It's when an organism possesses two factors. Factors is something very vague. We know they're talking about characteristics. So let's read it like how we know it. An organism possesses two characteristics which separate or segregate so that each gamete contains only one of these factors. So I'm going to bring you back to your mom and dad. So basically, a haploid sperm and a haploid ovum combine and they form a diploid zygote. And it undergoes meiosis, which is what they're talking about, where they separate or segregate. So that's what we're talking about here. And they form a haploid structure. So one allele from each parent is received by the organism. Okay, so that's what they're basically saying. You can see it here in the example. You have a variety of chromosomes, but you can see that the organism segregates so that each gamete contains only one of these factors. What is the one of each factor? One is maternal and the other one would be paternal. There you got it again, maternal and paternal. So they split up so that each factor is separated or segregated. Classic meiosis scenario here. Mendel's law of inheritance, there we got, in our diagram, we got brown eyes and we got blue eyes. And then we got the offspring we can see has brown eyes. But you need to remember that there is no competition here. It's everything that happens is very, very random. So when we're looking at this diagram, there is no, brown eyes doesn't have more of a chance to go into the offspring. Blue eyes doesn't have more chance. It's completely random. That is why when we say a couple is going to have a baby, it's 50-50. There's 50% chance of them having the baby. What law is this that we're talking about? We're talking about Mendel's law of independent assortment. So Mendel's law of independent assortment, independent meaning it does not need anything from another body, okay? Assortment means a mixture. So the various factors controlling different characteristics are separate entities, not influencing each other in any way and sorting themselves out independently during gamete formation. So here we have, let me use this as an example of two alleles. So here is the allele, the gene for eye color. Okay, so on this side the allele is blue, and on this side the allele is brown. There is no pressure from either allele that influences each other. It's completely random. So there's 50% chance that the person is going to have blue or brown eyes. The other thing that we can consider is maybe hair color. I'm just using color because it's easier to relate to. The hair color allele doesn't interfere with the eye color allele. That is what Mendel's law of independent assortment. It means that the alleles operate independently from each other. They don't influence each other. Okay? So that's Mendel's law of inheritance. But let's look at what we're going to do today. Monohybrid crosses. So when we look at the word mono meaning one and hybrid meaning mix, it's a mixture. Okay. So if we look at this, the definition of a monohybrid cross is a cross between two organisms. There must be two organisms, isn't it? There must be a mother and a father, or a male and a female. So every single cross that you're going to have two sets of gametes on here and two sets here, because one is coming from the mother and the other one is coming from the father. And you need to know when you are doing a monohybrid cross that you're always going to have four options. I mean, four possible outcomes, four possible offsprings, because we have gametes from the father and we have gametes from the mother. And as we are crossing them, we have four possible outcomes. And we also need to remember that the trait that we are studying in this diagram, in this case, it's color. It's the color of the pod, yellow and green. So when we are doing the monohybrid cross, we know the phenotype. That's the phenotype. And from the phenotype, we cross them with each other. And the genotype will reveal what is the offspring going to be. And in this case, we know that the offspring is going to be green. If we move on, this is a very important diagram. It's really, really important because you're going to get 2 marks for nothing here. They're just going to give it to you. But you need to learn the format of this, okay? So the format of the genetic cross. So there's 2 types of genetic crosses you can do. This one is called the line diagram because of all these lines, okay? But it's not my favorite because you can make a lot of mistakes with this cross, but you'll see why in a while. So when we're marking your grade 12 paper in paper 2, the first thing we do as a marker, we're going to look for P1 and F1. So as a marker, I'm going to look for P1, F1. Do you need to know the content? No, you don't need to know the content. You're just going to get a mark for writing P1, F1. Isn't life science great? Okay? We're giving you marks for nothing. And if you don't take it, then that's not good. Okay, so P1, what is P1? Obviously, P1 is the parent generation. F1, remember we said the parents have children and then they become a family. So F1 is basically the offspring. So you're going to get a mark. And obviously the parental generation, parents come before the offspring. So we're going to check that you know that the parents come before the offspring. So the parents must be on the top. and the offspring must be at the bottom, okay? And then we're going to look at phenotype. So P1 is going to be in line with phenotype. You know what phenotype is? Phenotype is the outward appearance. So you're going to have maybe a brown cow is crossed with a black cow, something like that. So the male is going to be put on the one side and the cross to indicate that they are being crossed. And then the female is going to occur on the other side, okay? And then there's the genotype. The genotype, you're going to have letters, isn't it? You're going to have a capital letter or a small letter. They're either going to be homozygous or heterozygous. But in the phenotype, they're going to tell you what it's going to be. So from the genotype, in between the genotype and the gametes, you're going to have meiosis. So the position of meiosis is very significant because from the genotype, those genes are going to undergo meiosis and they're going to form gametes. So, for example, if you got capital B here, capital B, they're going to separate and they're going to form gametes there. Do you see? And the same for the female. And then these gametes are going to be crossed with each other. And then when they cross with each other or when they fuse, we know that this is called fertilization. We need to use the term fertilization. So meiosis cannot happen after fertilization. Now, lots of learners feel, oh, I'm just going to put fertilization there and I'm going to put meiosis afterwards. It's going to be marked wrong. When I'm marking that paper, I'm going to check. Meiosis, is it first? Yes. Where is meiosis? It's between the genotype and the phenotype. And fertilization, okay, sharp. I'm going to give this learner a mark. Okay? So that is how we mark the papers. And then once you've done the cross, we're going to do a cross just now, but I just want you to know the format of it. Then you're going to do the genotype, the outcome of this. So you're going to get results here, and then you're going to do the genotype there and the phenotype. So you're going to write the letters here, and then you're going to write the phenotype, which is a description of what is actually going on. And also, you must be very careful because a lot of questions ask for the ratio. So if they do ask for the ratio, even if they don't ask for the ratio, rather put the ratio in and work out the ratio in its simplest form, just to be sure. So here at the bottom, we've indicated what you're getting marks for, but that is just by the way, we've already discussed it. So that is the genetic cross. So that is the line genetic cross. And this is problematic. I wouldn't encourage you to do this because when you are doing this crossing and all of this going that way, from that male to that male, from that female to that female, male to female, you're going to become confused with the lines. There's so much room for error. I would recommend that you actually use the Punnett square. But we're going to come to the Punnett square now. We're going to have a quick run through as a summary of what we've just done. The visible trait is the phenotype. There's the phenotype, isn't it? You'll find it there. The genetic makeup is the genotype. So you're going to find the letters there. Do you remember I told you how you need to know the definitions? You need to know the terminology. See, I wasn't lying. There's it. Okay? So you already know this. So you're going to be okay. The next part is the alleles segregate to separate during meiosis. There it is. It's separating, okay? And during fertilization, the individual gets one allele from each parent. You can see, because of that cross, it's going to get one allele from each parent, okay? So this is the format of the genetic cross that is quite nice to use. It has the same format that we discussed before, the P1, the F1, the meiosis and the fertilization, the gametes being formed, but very importantly, the Punnett square. So the Punnett square gives you an opportunity to check that you didn't leave anything out and you didn't forget a cross. Okay? So that is the format of the genetic cross with the Punnett square. Remember that by writing P1, F1 and meiosis in the correct sequence, see what they say? Correct sequence. You can't just write it for the sake of writing it. It must be in the exact same row and then you will get marks. So there's lots of marks to be gotten here. And you must just take advantage of the opportunities presented to you. So let us look at complete dominance. Just want to go back a little bit. Yes. Okay, so we're going to look at complete dominance. So we're going to quickly run through examples because yes, we've learned the work, but we need to know through examples because you can't just learn content and not do examples because that's not really exercising your knowledge of the subject. So here we have complete dominance, okay? So there we have red, we've got the red flower crossed with a white flower, okay? So the red is homozygous dominant. Homozygous meaning the same, dominant because there's two capital letters. And then you got homozygous, we got homozygous recessive, which is the white. But if you look at this diagram, you can't see any white here. So you can see there is complete dominance. This is where Mendel's law of dominance is actually shown quite nicely. So here we have if you look at the offspring, even though they are heterozygous, we cannot see the red there. That is complete dominance, showing Mendel's law of dominance. Let us move on and read the bubble. And a genetic interaction where one allele of a gene suppresses the expression of an alternative allele in the F1 heterozygote, example BB, so that the phenotype is the same as that of the dominant allele. All they're saying there is that the phenotype will reflect red, and it will have white, but we can't see the white. The white is in the genotype, but it's not expressed in the phenotype. Let's talk about complete dominance. So there are 4 types of complete dominance that can take place. So 4 types of crosses that can be carried out in complete dominance. Homozygous dominant times homozygous recessive, heterozygous crossed with heterozygous, homozygous dominant crossed with heterozygous, homozygous recessive, and heterozygous. So we're going to use these cute little bunnies as our example here. So in rabbits, the allele for black fur is capital B. That's what we're going to be using. It's dominant. We can already see that it's dominant. They didn't need to tell us that because we can see it's capital letter over the allele of white fur, which is B. Okay, so it's going to be black fur and white fur. So here is a worked out example of how you're going to do a genetic cross where it's homozygous dominant crossed with homozygous recessive. So if they ask you in the exam, they say cross homozygous dominant with homozygous recessive, they just give this to you. Okay, they'll give you a little other details, but from that already you'll be able to take out information. You will see homozygous. Then you already know it's going to be 2 same alphabets. You see dominant. You're going to know that those alphabets are going to be upper caps. It's going to be capital letters. There you go. And it's going to be black. They're not telling you it's black. But when we work out the genotype and we look at the description, we are able to work out the genotype. Okay? There, homozygous. Homozygous meaning 2 same letters, recessive. And we know we're dealing, in the question they told us, small b is recessive. And there we got it, BB. Then they undergo meiosis, and as you can see, the gametes are formed. Can you see that? The gametes are formed, and the gametes undergo fertilization. There you have the Punnett square. The capital B is put in there. The next one is put in there. The small b is put in on the left-hand side, and the other small b is put on the left-hand side. And then we combine them. So we take the capital B and the small b, and we get that. Capital B and the small b, and then we get that. And we do the same. Capital B, small b. I sound like I'm repeating myself. I am. Capital B and small b. And there we go. And we already can see. We don't even have to look at the answer. We know that it's going to be heterozygous. And we can see that there's going to be capital letter B. So it's going to be heterozygous dominant for black. The F1, remember we said it's the first offspring. And then we're going to take the common letters here. Remember, each block represents 25%. So each block, for example, that BB is 1 out of 4, which is 25%. Okay? And then you have the genotype, which is expressed. So all of these are BB. So we don't have to write BB, BB. We summarize it and we say that the genotype is going to be BB. Okay, so then we have the phenotype, which is going to be expressed as we look at the genotype. I'm just going to run through the next one. We have heterozygous and heterozygous. Very, very clear there. Heterozygous, you know that you're going to get 2 letters. There's the 2 letters. And then we have the gametes forming, the Punnett square. very important, what I want you to pay attention to is the ratio. Okay, so we know that we've got one capital B, and two capital B small b's, and two small b's. Okay, but we know that ratios need to be always written in its simplest form, and that is how we get 3:1. The phenotype will be reflected as 75% black. How do we know it's 75%? Remember, we mentioned 25% represents each block in the Punnett square. And then finally, we're going to do homozygous dominant and heterozygous dominant. So that's where black rabbit is going to be crossed with the black rabbit. And because this is heterozygous, these are going to be two different letters. Can you see that? It's becoming so easy now that we know our definitions. And then very important step, the gametes are formed. And there you have the Punnett square, which you will summarize at the bottom and write down the ratio 1:1, which is black. So grade 12s, thank you so much for your attention. I encourage you to stay tuned. We're going to give you a little bit of a break to freshen up. My colleague Agnes is going to join you in just a few minutes. Thank you so much.
Attitude of gratitude. What does that mean? An attitude of gratitude is the quality of being thankful. It's the quality of giving thanks. Ubonga, ulebuha. It's to be grateful. You and I have so much to be grateful for. You and I have parents who make sacrifices day and night for us to be at school. You and I have people who believe in us when we don't believe in ourselves, who love us when we don't love ourselves, who invest in us day and time again. Whether it's a teacher, it's a principal, it's a driver, it's a guardian, we have so much to be grateful for. And so I challenge you, young person, be thankful, ubonge, lebuha, be grateful each and every single day of your life. Strive to display an attitude of gratitude. Excellence. Excellence is a habit. Excellence is a quality you have, a quality I have. It's a quality we have when we honor what is true to us. Yes, we are gifted differently. uNkulunkulu akasinganga ngokufana. Excellence is striving for your best. Whenever you have a particular project, test, homework, uzibuze, ask yourself, is this my best? Can I put in more effort? Excellence is striving to be in the top 1% of the area in which you are gifted. And so I challenge you, in every single thing that you do, in every single area that you're gifted in, whether it's sports, it's academic, ucula, whatever that gift is, strive for excellence.
Look at where you have been and ask yourself, am I happy with the results? Look at where you are right now and ask yourself, you have been given power to decide whether you want good results in your life, in your schoolwork, or you want bad results. As Tony Robbins said, it is in the moment of your decision that your destiny is shaped. And your destiny is shaped right now with the decisions that you are currently making in your schoolwork. So decide whether you want to be an A student and start working on it, because there is power inside of you. The infinite power is inside of you, and the time you decide to tap in that power, you will become who you want to become in your schoolwork.
Thank you, Samantha, for the wonderful session one that we had with the grade 12 learners. And good afternoon, grade 12 learners. My name is Agnes, and I'm going to take you through the answer and question session. Now, as Samantha has mentioned, with genetics, it carries most of the marks, 48 marks of paper 2. So if you score all the marks there are greater chances that you're going to pass your paper 2. Now, the questions that we are going to start with comes from November 2018. This is our paper 2. Now, one tip that I'm going to share with you is that before you can attempt any question, you first have to do what? To read with understanding, read the given information, then look at the mark allocation, then attempt. Right, let's go through the questions together. A flower, purple or white in a particular plant species, can I repeat that? Flower color that is purple or white in a particular plant species is controlled by 2 alleles, capital D and small letter d. 4 crosses were carried out to determine which allele is dominant. Remember, we spoke about the dominant allele, the recessive allele, in our first session. So 40 offspring were produced in each cross. The phenotypes of the parents and offspring in each cross were recorded. Now, if we look at the diagram given there, we were told in the above information that we have got 4 crosses that took place. And here on the phenotypes, we've got parent 1 crossing with parent 2, and they give us the offspring. Now, before you can answer, also study that diagram quickly. Remember, 1 minute is equal to 1 mark, and you need to budget your time profitably so. So it's purple crossing with white, they give us 40 purple. Purple and purple, they give us 31 purple and 9 white. White crossing with a white, they give us 40 white. And then purple with white crossing with white, they give us 21 purple and 19 white. Now, if you can check, all the offspring here, many of them are of purple color. Now, the first question says, state the dominant flower. So now if you can look at your table there, the dominant color is what? Is purple, and this is for 1 mark. Like if the question requires 1 mark, it's straightforward. You just write the answer and do not rewrite the question. Question number 2, use cross 1 to explain your answer to question 1. Now use cross 1. This is cross 1. Now if you go back to cross 1, is purple crossing with white. We got what? 40 purple as our offspring. Now the answer says when purple and white flowers, plants are crossed, this is a mark, all offspring has a purple flower. Or looking at the table again, with the offspring we have 40 purple. You can just say there's no white flower there. That is how you need to approach the answer. Now, state number 3, state the Mendel's law of segregation, as Samantha has already done it with you to explain the concepts. Like with the law of segregation or with any definition that you have in life sciences, please make sure if you have to memorize, you can memorize, but with understanding so that you don't forget. Now state the Mendel's law of segregation for 3 marks. So it says an organism possesses 2 factors which separate or segregate. Remember, the word segregation means what? To separate. So the Mendel's law of segregation states that an organism possesses 2 factors which separate or segregate, and then so that each gamete consists only of what? 1 of these factors. Then you get marks for stating that it possesses 2 factors, it's a mark, and for then for you to show that there's separation taking place, there's segregation taking place, it's a mark. The results of that is that each gamete will contain only one of those factors. That is 3 marks. Now the next question, because remember with genetics, you were taught during the first 3 weeks of this term that we have a monohybrid cross, we have a dihybrid cross. Now let us look at this type of a cross. Number 4, it says use a genetic cross to show how crossing of 2 purple flowering plants can produce white offspring, as in cross 2. Remember, we spoke about 4 crosses there. We had cross 1, 2, 3, 4. Now, in our second question, we needed to do what? We used cross 1 to explain how dominant flowers can be produced, how dominant parents can produce offspring with those specific colors. Now, in this number 4, we say use genetic cross to show how the crossing of 2 purple flowering plants can produce white flowering offspring, as in 2. This is 2, purple and purple. They gave us 31 purple and 8 white. Right. Now, this is the wet example. Remember, when you do a monohybrid cross, what is also important is that, as Samantha has mentioned, you need to show the phenotype and the genotype of the P1. Now, what you need to do, you first write your P1 there, and then for your P1, you write the phenotype and the genotype. Listen, don't confuse the two. Remember, with the phenotype, that is the external appearance. With the phenotype, you're talking about what you see, the outside appearance. So the phenotype of P1, remember we were talking about what? Cross number 2, a purple crossing with a purple. Now, I want you to listen very carefully. You need to show that purple crosses with purple. So if you don't write this sign to indicate that crossing is taking place, you are going to lose marks in your exams. So we need to see that you understand what crossing means. So purple crossing with purple, it's a mark, if you can check. And then the genotype. The genotype is the makeup composition. So with genotype you can't see, but with phenotype you can see the characteristic. Now the genotype for purple, as indicated in the information given there. Purple is represented by capital D and small letter D, which shows the what? The dominancy here. And then crossing with what? Purple flower, capital D and small letter D. And as you can see, for you to write the phenotype and the genotype, you already have what? You've got 2 marks allocated to you. Then, with these alleles, what is going to happen? Meiosis is going to take place. Why do we need to have meiosis taking place there? In order to separate the gametes. Can you see those? So meiosis first take place to separate the gametes of one parent, to separate the gametes of the other one. Why do we have separation of the gametes? So that fertilization can take place. Now let's check. When fertilization has taken place, remember in the first session we spoke about the order of the positioning of meiosis and fertilization. We first need to have meiosis taking place to separate the gametes. Now when gametes are being separated, then fertilization will take place. Now when fertilization takes place, what are we expecting? We are expecting to get the results. In this case, we need to see our offspring. Now, when you do the diagrammatic presentation or the Punnett square, you need to indicate that now after fertilization, we're expecting what? Our first generation, which are the offspring. Now, it's important also that with the F1, you indicate the genotype and the phenotype of the F1. So before we go to that last part, remember, crossing has taken place. You have indicated with the cross to indicate that purple is crossing with purple. Gametes were separated so that fertilization takes place. So as a guide, now if you do write a Punnett square, you can write if in your scenario is depicting or is indicating that this is male, this is female, it's advisable to write your gametes, your male gametes on top here, and then the female gametes on the side, right? So we have D, 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 D. Here are they, the D and the D and the D and the D. So to make sure that you don't mess up, because these are free marks, we usually talk about low-hanging fruits. Now with the low-hanging fruits, it's easy for grade 12 learners to just get. If this is a tree with fruits hanging below or hanging down, you just pick up, get those marks free. Right. Now with this one, in many times it's for 6 marks. It's important to get those 6 marks. This we call them free marks. Right. So how do you do that? How do you get your offspring? When fertilization takes place, what are we expecting to happen to the gametes? So you say D fuses with D, they give us what? DD. The first D fuses with the second D. This is what we get. Now you come again to say this one fuses with this one is a DD. So it is advisable to start with a capital letter if we've got capital letter and a small letter, right? So this small letter and capital letter D, they give us DD. And then this small letter D fusing with this letter D, we're going to get small letter d. Now we call those homozygous recessive. Okay, the last ones. Right. Now, after fertilization has taken place, you need to come up with the genotype of the F1. Now, you read the results. You read the offspring which are in here. So, it will be, the first one is capital D, here is it. Then, small letter, capital D, small letter d, here is it. And then the other one, here is it. I hope you all see that. And then we've got a small letter d's, here they are. Then because we have the results there, we have our genotype there, then we're going to write the phenotype. Now with the phenotype, remember you are describing what you see. So this represents purple, this represents purple, DD represents purple, the small d and small d represents white. Then you need to read all those results to say you have got how many purples, because we've got many purples there. You literally count there, it's 1, 2, 3. 3 purple. And how many white do we have? We've got 1. I hope you understand. So that is why I was saying with this one, make sure that you have your P1 and F1. You can even indicate that you understand or you have understood when you were taught. So for you to write the correct position of P1 and F1, you get a mark. For you to indicate that meiosis is responsible for separation of the gametes so that fertilization takes place. If the correct position is there for meiosis followed by the gametes and fertilization, you can even write this in your exams so that we give you a tick. So it's free 2 marks here. Then you can get any other mark from separation, from you getting the correct alleles, correct gametes, and then fertilization taking place, and also getting the F1 and the genotype and phenotype of the F1 in the correct positions, right? Now we're going to talk about complete dominance and codominance. Yes, we have spoken about this in our first session. Now with the incomplete dominance, what does it mean? Right, for an example, in this case, I've got a red flower, I've got a white flower. Now, the alleles there for red is capital RR, 2 capital RR, and for white is 2 capital WW. Now, what will happen if the red flower crosses with the white flower? Check. We've got a different color, which is pink. Now, in this case, remember, a red flower crossing with a white flower, we're getting a pink color. Look at the alleles there. We've got capital RR for red, WW for white, and then we've got RW for the pink. So, pink in this case shows intermediate phenotype. Now, when you think or when you see the word incomplete dominance in your mind, think of what? Two things crossing, or crossing between, in this case, crossing between different colors, flowers of different colors. Incomplete dominance, think of intermediate phenotype. So neither red nor white is seen in this intermediate phenotype. So incomplete dominance, neither one of the two alleles of a gene is dominant over the other. None of them is dominant over the other. Hence, the results, we have got what? We have got intermediate phenotype. In what? In the heterozygous condition. So we've got R, capital RW, and capital W there. So this means neither one of the two alleles of a gene is dominant over the other. Right? Let's compare incomplete dominance with codominance. Ask yourself, what does co mean? Co, in simpler way, can mean both, can mean together. So codominance means what? Here we are given an example of a red flower crossing with a white flower. Now let's look at the alleles there. We've got capital RR and capital WW for white. So in this case where we've got red crossing with white, remember we are comparing incomplete dominance and codominance. Now in this case, we see the results here. The flower has got colors from both the flowers. So we've got red, you can see the red color on the offspring, you can see the white color on the offspring. So in summary, with codominance, we are saying both alleles of a gene are equally dominant. They are equally dominant. Hence, we see both colors in the offspring. Right. Or what you can say is alleles express themselves in the phenotype in the heterozygous condition. So in summary, codominance, both alleles of a gene are equally dominant, whereby both alleles express themselves in the phenotype in the heterozygous condition. So when you see these terms in your exams, do not be surprised. Link the incomplete dominance with the intermediate phenotype, where neither of the colors will be represented in the offspring. With the codominance, think of what? Think of the result, the offspring, whereby you can see both colors dominating there. Right? These are examples of the incomplete dominance and the codominance. Now we are representing that in a cross, in a genetic cross. All right? So remember, when you do the genetic cross, you have a choice. You can use diagrammatic representation. You have a choice. You can use the Punnett square. It's up to you. The one that works for you, if you want to forget, the better. Right? Now, for incomplete dominance, Remember we spoke about the red and the white flower. Don't forget the cross must be there to show that this is a cross, this is a genetic cross, is a tick. Genotype of the P1 is important. If you forget to write P1, you are going to lose marks as well here. Now, it is given in the information that red is represented by capital RR, and then the white is represented by capital WW, then this is a mark. Then remember, in order for these gametes to separate, meiosis must take place. Then when meiosis takes place, it separates the gametes. Girls and boys, grade 12 learners, do not make a mistake of leaving spaces in between these cells. Do not leave spaces. But immediately when meiosis has taken place to form the gametes, you can leave a space to show that these are separate gametes. Can you see? These R and R. You can write a comma so that we know they are separated. Now they are crossing. You can leave a space to show that we've got separate gametes. All right? Now fertilization takes place. You can take the RR on top and then the WW on the side here. Then you cross W and R, you get RW. You cross W and this R, you get another RW. You come with the second one, W and R, you get RW, and then the second one with this one. All right, to get RW. Now, it's easy for you when you have represented in this way to get the F1. Now, with the F1, you literally count what you have in here. So the genotype, you'll be having RW, you rewrite it here, comma, RW, you rewrite it here, RW, you rewrite it here. Then you give the phenotype of the genotype of the F1. So if you read the whole results here, what are we getting? We're getting 1, 2, 3, 4. All flowers are pink. So it means when we say 100%, it means all of them. Remember, we can read this as 100. This is 25, 25, 25, 25. This will give us 100. So 100% of the flowers are pink. That is incomplete dominance. In this case called dominance, we were given red and white. The phenotype here, we've got a red and white is a mark. Then the genotype, RR, we've got WW. They are given, by the way. Those will be given to you. It's a tick. And then meiosis take place so that gametes are separated and fertilization takes place. You get a mark. Now for you, in order for showing us that you understand that fertilization has taken place, you will get a mark for the gametes. You will get a mark for showing that fertilization has taken place. W and R is RW. W and R is RW. Can you see that? And then W and R will give us RW. W and this R will give us RW. Then you rewrite those offspring again as your genotype of your F1. So this will give us all of this. And then we've got red and white flowers in this case. That is our phenotype. So what is important again? P1, F1, for you to qualify for a mark, meiosis and fertilization position must be correct. You get a mark, then you can get marks anywhere here. What is also important, you need to read the question, what it requires. You don't just make a cross without understanding at the end of the genetic cross what is expected of you. Read the whole question up to the end. Right. The next question that we're going to practice on is 2021 May-June paper 2. Hence, it is important that you make a collection of those past papers. Make a collection for yourself. Take those questions. Drill yourself, answer. Get those, answer those questions. You'll get the memos as well. Your teachers will provide you with that. Or you can just, you've got many resources for that. Now, get that collection, start to practice, because it is possible that whatever you're going to practice on may come out in the June exam, prelim exam, and end of the year exam. Right? Even if they don't come out, but you'll be having a way of attempting the questions. You'll be getting used to how we ask questions in life sciences. Right. Here is the next question. In bunnies, their color may be black, white, or gray. The inheritance of fur color is controlled by two alleles, namely, for black fur is capital V, and for white fur is capital W. Explain why color in bunnies is an example of inheritance with incomplete dominance. Remember, I spoke about incomplete dominance. What should it tell you? I spoke about codominance, what you need to think about when you see those words, right? Explain why fur color in bunnies is an example of inheritance with incomplete dominance for 2 marks, right? You are even given the alleles there. Don't forget. So everything is given. Yours is just to understand the question, then attempt. Right. Explain why fur color in bunnies is an example of inheritance with incomplete dominance. So the gray color is an intermediate phenotype. Remember, we spoke about that. When you see the word incomplete dominance, think about what? Intermediate phenotype. Or you can just say, the other way of understanding what intermediate phenotype is, you can say it's a blend of black and white. It's a mark there. Remember, it was for 2 marks. This indicates that neither of the alleles is dominant. Remember, with incomplete dominance, we're talking about what? Intermediate phenotype, we're talking about what? Neither of the alleles is dominant. That is incomplete dominance. I hope you get that right. Now, the next question, number 2, use a genetic cross to show the expected genotypes and phenotypes of the offspring when a gray male mates with a black male. Mating there refers to crossing. Okay? Right? When a gray mates with a black. P1, genotype and phenotype. Okay? So this is the external appearance. Phenotype, external appearance is a mark. And then for you to get the genotypes right, it's a mark. So, girls and boys, if you can get the P1 correct, I want to believe that the whole workings there will be right. But if you can mess up your P1, then everything's going to be wrong. So make sure that you understand the question so that you get your P1 correct. Right? Now for gray, we've got B, capital letter B and capital letter W. For black, capital letter B, B. Right? Meiosis takes place to separate the gametes. We've got, they are separating, by the way. As they are separating, we've got B, we've got W, they are separating. BB, they are separating. Can you see that? Right, then fertilization takes place. Now, when fertilization takes place, these are the results. We've got BW gametes fusing with BB gametes. This is what we've got. Now, first, F1 genotype and phenotype. Now, the genotype, we've got BB. Remember, let me show you for the last time how we crossing these gametes. So this B1 will cross with this to give us BB. Okay, are we still together? And this one will cross with this one. We get BW. Then this B crosses with this first one. We get BB caps. And then this one crosses with this. We get BW. I hope you get that right. Then what is inside? Remember, these are gametes. These are gametes. Then you write the genotype that you get in this. Make sure that your box, you draw the correct box. We don't just want a cross there. All right. So this is BB. This is the next BB. This is BW. This is BW. So if we look at the phenotype of the four, BB is black. Phenotype is what you see, is the external appearance. Now with the phenotype, think of what? External appearance. You can see I'm brown. You can see I'm tall. So that's what you see, right? B is black. BB is black, right? And then BW is gray. BW is gray. So in terms of the phenotype, what do we have there? We have got 1, 2, 2 black. We've got 1, 2, 2 gray. I hope this will do. Not forgetting P1 and F1, meiosis and fertilization. So you're halfway to your 48 marks of paper 2, right? The next activity, we've got a farmer has an orchard of apple trees. Each apple produced expressed red and yellow color equally. That is red, yellow apples. To extend his apple orchard, the farmer collected seeds from the red, yellow apples and grew them. When the new trees matured, he found that some of the trees produced red apples. Now the red apples are represented by what? Capital letter R. Others produced yellow apples. Now the yellow apples are represented by what? Capital letter Y. And the rest produced apples that were red, yellow, right? So read with understanding, expecting that questions are coming from what is given. Right. Use a genetic cross to explain his results in the F1 generation. Like I've indicated earlier on that for genetic cross, 6 marks are enough. And I've shown you where you can get those free marks. Right. This is the cross whereby we said P1. We have red, yellow crossing with red, yellow. Remember, a farmer needed what? Red, yellow apples. So in order for the farmer to get more red, yellow apples, what should he do? Cross red, yellow with red, yellow. That is a mark. And then the genotype, you were given the alleles there. So for red, yellow is R and Y capital letters, crossing with red, yellow, R and Y capital letters. Okay? Then meiosis takes place. We have separation of the gametes. Can you see? That RY, they are separated now. You need to show us that you understand by leaving a space in between so that you understand that gametes have separated. Okay? Then fertilization takes place. I also love the diagrammatic representation because it will bring that concentration so that, you get all of those correct. So what is going to happen? You have separated the red, yellow gametes. Fertilization takes place. The first one with the first one of that parent is RR. That's how we read it. Because if you leave this open, space open here for your offspring, you're going to lose the marks there, right? Then the first one again with the first one of the other gamete is RY. Then this one with this one is RY. The last one is RY. Also, for you to get the genotype correct, you get a mark. Then you interpret the genotype in terms of what you see. What are your results? What is the phenotype there? RR means red. So this is red apples because we're talking about apples. And then we've got RY, we've got RI. RY means we've got red, yellow apples. There are two, don't forget, is RY and RY. Then the last one we have got YY. This is yellow apples. So the results here, we've got red apples, we've got red 2, this is 1, this means 1. If you don't have a number here, then we've got 2 red yellow apples, then we've got 1 yellow apples. So don't forget, for you to get P1, F1, to get meiosis and fertilization. Then you get your full marks there. Alright? So look at this question. What proportion of apples in the F generation will be red yellow apples? Now the question says, what proportion? So it means if the question is asked in this way, you need to do your genetic cross, but then this is not the end. You can't end where? On F1 generation, because the question needs you to go further. It's asking you further to explain what proportion, and don't confuse proportion with the ratio. Here it says what proportion of apples in the F1 generation will be red yellow. Now I have done it for you already to say the red yellow we've got 2. Remember I've said this represents 100%. So this will be 2 out of 4. Now 2 out of 4 is half, and a half means what? 50. Half of 100 is 50, so it's 50%. So the question says, what proportion of apples in the F1 generation will be red yellow apples? The proportion is we are going to get 50% of red yellow apples. So read your questions up to the end so that you make sure that you answer everything. Now immediately when they say what proportion, it means you have to get that mark correct. And if you get it correct, you get full marks. But if you miss the end of the question, then you are not going to get full marks. Right? The next activity continues with the farmer getting the red yellow apples. Remember we did question 1 and 2 in the previous page. Now in this page we're doing what? The farmer saw that the red yellow apples sell the best. And if you are a farmer and it's selling the best, what is the best thing to do? You cross red yellow apples with red yellow apples in order for you to make money, right? Name the phenotypes of the trees that should cross in the future in order to ensure that any new trees will definitely produce only red yellow apples. Name the phenotype. Right. Now if you name the phenotype, remember that question was for 6 marks. So we continue with this one for 2 marks. A tree with red apples should be crossed with a tree with red apples so that it produces what? Yellow apples. Remember you need more so that you sell, so that you make money. So you must cross the red apples with red apples so that you get, it produces more yellow apples. Is that right? Okay, let's move on. All right, sex determination. We just left with few minutes to complete. Please remain tuned. Right, sex determination. Now, it's easier when we look at the phenotype. I can tell that you are a boy. I can tell that you are a girl. You can tell that I'm a woman, right? So, what makes us to be like that? What makes you to be a boy? What makes you to be a girl? Right, in humans, don't forget, we've got the body chromosomes, we've got the sex chromosomes. So all in all, the human body has got 46 6 chromosomes, of which 44 chromosomes are body chromosomes, and we call those the autosomes, right? So you can either say the first chromosome up to 22 pairs, 1 to 22 pairs, they are body chromosomes. We call those autosomes. Then the last pair, which is on the 23rd pair, which is a sex chromosome, we call those what? The gonosomes. So how do we differentiate between a boy and a girl, right? So males, for an example, they have got XY in their 23rd chromosomes. Females have got XX in their 23rd pair of chromosomes, right? So we've got 44 autosomes, we've got last pair of what? Of chromosomes. Right? Now, just to cover up the last one, for us to determine the sex of an individual, a male has got 23rd pair, which is XX, and last pair, which is XY. So males have got XY, females have got XX. So in order to wrap it out, just for me to finish up, you know, in our everyday life, men or our fathers will tell our mothers, men will tell the wife to say, wifey, give me 2 boys, I want 5 boys. Is that possible? No. No one can determine the sex of the offspring, the sex of the offspring or babies. So the male will have that sperm cell. Males have got 2 types of sperm cells. Sperm cell with what? 22 and X chromosome. Sperm cells with what? 22 and Y chromosome. So what is going to happen? The first sperm that will fertilize the ova is the one that is going to determine whether you're going to get a boy or a girl. So I was going to do more, I will do more examples in the next session, because my time is up. And good luck, grade 12 learners, as you will be writing the June exam on the 5th of June. Good luck. Make sure that you master all the content for term 1 and term 2, right? Goodbye and study well. Thank you.